The eyeball is very fascinating, isn't it? I have to have corrective specs. That's something that I may try to fix one day using our awesome technology and laser surgery. But I'm very curious about why I can't focus on images properly if I don't have lenses. But I can understand a little bit. Here is the general structure of the eye. You should be able to label an eye diagram with all these various things that are here. Chances are you probably learned this at some point in your scientific studies. So just quickly, these are some of the names here, and then we'll go over some of the details. But we're specifically talking about how we can actually process the light image. And we're also going to be talking in another video about various ways that we process various types of visual stimuli. The eye is really interesting in how it interacts with the brain. Optical illusions are kind of cool to look at, but they're evidence that our brains and eyes are kind of stupid because they can be tricked really easily like that. There's a lot of problems with the design of the eye. So the lens, of course, helps to focus some of the light there. Aqueous humor, pupil is the hole where light goes through. Iris gives us the color of our eyes. Everybody's iris is unique, so we can do retinal scans and stuff like that. Conjunctiva, you probably heard of in terms of, it's an outer layer, but conjunctivitis is when you can get inflammation of that particular layer. The cornea, vitreous humor, sclera is a tough outer layer. If you do any kind of eyeball dissection, this is a really tough layer. You have to actually pierce that with scissors or a scalpel. It's kind of fun, but a little bit scary. Choroid, we're going to talk about later. The retina is the most active part. That's where all the photo absorbing, the light absorbing layer is, and we're going to talk about rod and cone cells. The fovea is the focus point. The point of the lens is to take the light rays that come in and actually focus it onto this point. Anything that shows up here is what you're interpreting as what you're directly staring at, so you get a very sharp, clear image there. Blind spot is where the optic nerve actually connects, and this connects onto your brain. There are no, you notice here, there's actually a gap. There are no rod and cone cells for detecting light here. So any light that actually falls here, you can't see. And so you can try a little blind spot activity. Just search for it online or in your textbook, and it's really, really fun to do. And you don't realize that when you're staring straight at something, there's particular parts in your field of vision that have completely disappeared, and so you may not notice certain things that are there. Your brain is really good at filling in some of the gaps, though. So, okay, let's take a look at the structure of the retina, and we'll see how this overlays in a second. Well, here's a large diagram showing this. So if you take a section out, here's what we're looking at. These are the photosensitive neurons, these rods and cone cells back here. The light is going through this direction, and then, but in front of it, there's all these bipolar neurons, everything that's actually connecting these all up into your optic nerve. We're going to see that in a second here. So when we're taking a look at the photosensitive layer, the direction of light is coming in, and you see these oddly shaped cells that look like little combs to keep your hair nice and neat. You have a layer of pigmented cells back here. These, it's actually black. The layer is actually black here, and so any light that comes through here is supposed to be picked up by these rods and cone cells, but anything that doesn't get picked up will land on this back layer, and it's black so that that light actually gets absorbed. If it were shiny, then some of this information could bounce off, then come back this way. Then we'd get really strange images being processed by our brain, and we wouldn't be able to cope with that. Cone cell is a type of photoreceptor cell. We have 3 different types. Rod cell is another type of photoreceptor cell. Immediately you can see some differences, and you have to be able to distinguish between cone cells and rod cells later on. So the 3 types of cone cells are responsible for color vision. They detect red wavelengths, green wavelengths, and blue wavelengths. The rod cells are more for kind of black and white vision. These are what help us to see when we turn off the lights immediately, and then we start to get accustomed to looking and seeing in the dark. We don't detect colors very well in the dark, right? But it's the rod cells that are kicking in to actually do that. Bipolar neurons are going to transfer that information from these cone cells and rod cells to these particular special ganglion neurons that are going to connect up to the optic nerve. So you'll notice here that a group of rod cells, so this tells you that individually they're less sensitive. A group of them actually connects up into a single neuron. So as opposed to a cone cell, where a cone cell, an individual cone cell has its own individual neuron. So you get a lot more information from here, and we're going to see why these are more important and when we do a direct comparison between them. So if you actually compare the cone cells and the rod cells, here's a nice table that summarizes everything. Cone cells are primarily found in the fovea. That's your focusing point. They're not distributed much outside of the fovea. So when you stare at something directly, when you're staring at somebody's face, you are concentrating that light to the fovea, basically. Rods are found outside of the fovea. I've already mentioned they're used for night vision or achromatic vision, so not with color. Cones are used in bright light for day vision to help us detect colors. Several rods are attached to one bipolar neuron, whereas cones have individual bipolar neurons per cone. The accuracy is less accurate because of this several being attached to one bipolar neuron as opposed to cones. How many types? There's only one type of rod. There are 3 types of cones, and the cones are sensitive to red, green, or blue. Rods are sensitive to all colors at less, what am I trying to say? Sensitivity, I guess. Type of vision, black and white and rods versus color vision with cones, and many more rods exist than the cones actually. And here are some beautiful pictures showing some artist interpretation. So cones obviously less frequent than rods, and then you can see the difference in their actual structure, but you don't need to know their specific structure. Know that there's a lot of mitochondria that are actually there to help with that. So that's a quick summary of the eye and its structure and its functions and different parts that help us to actually detect light. Next, we'll be moving on to specific aspects of processing visual stimuli.